What is up, you guys? Welcome to Controversial Thoughts with Carnivore MD. In case you didn't know, this is my birthday. I am recording this and researching this on my birthday because it's so interesting to me. This is what I do on my birthday, so let's jump into it. I have been talking a lot about polyunsaturated fatty acids recently. Wanted to break down some interesting experiments that I've seen, and I'll be talking a lot more about this in the near future. And at the end of this video, I will give my thoughts on coronavirus and a little bit on the recent piece in The Guardian about not eating meat. But you guys should check out the podcast that is coming tonight. It'll be released tomorrow with Rob Wolf, Diana Rogers. I interviewed them to talk about their new book, Sacred Cow. It's a super interesting, hard-hitting interview about why we've been so misled regarding the environment, and so many pieces of really just frankly incorrect information have been widely promulgated. So check out that information on the podcast coming tonight and tomorrow. Please check out my book, The Carnivore Code. There's the first edition. Second edition is coming August the 4th, The Carnivore Code book right now to pre-order. Let's jump into some really cool stuff. I want to show you guys a series of experiments in mice, and then an experiment in humans involving both linoleic acid and stearic acid. And I'll talk a little bit about palmitic acid too. Now, in case you are not familiar with what we are talking about here with these fatty acids, we are talking about saturated and unsaturated fatty acids. I am talking about stearic acid, which is an 18-carbon saturated fatty acid, which has some pretty darn interesting effects in both mice, murine, and human physiology. We'll talk a little bit about palmitic acid, and I'll talk more about this one in the future. And then we're talking about linoleic acid, which is this 18-carbon omega-6 with 2 unsaturation points. So those are the main fatty acids I'm going to be talking about today, and how they affect things differently in both mice and human biology. So let's show a couple of really interesting mouse studies that have to do with this. The first mouse study is a pretty old one. It's a really interesting one, though. And this is one that I've talked about with Ivor Cummins on a previous podcast I did with him. The title is Dietary Linoleic Acid. So this is an 18-carbon polyunsaturated fatty acid is required for the development of experimentally induced alcoholic liver injury. And this is in a rat model. You can see male Wistar rats fed a nutritionally adequate diet with ethanol. So basically they made these rats into alcoholics. And we know that in humans, alcohol will lead to alcoholic liver injury. And what they did was really interesting. They fed the rats fat as 25% of the calories, either as tallow, which I would say is amazing, but I'll let you guys draw your own conclusions, which is rendered animal fat, generally from suet, which I think is really cool. People have heard me talk about suet before. And that is 0.7% linoleic acid. They fed the rats 25% of calories as lard, which was 2.5 linoleic acid, or tallow supplemented with linoleic acid at 2.5%. They then looked to see liver damage, and what they found was that the tallow ethanol group did not show any evidence of liver injury with alcohol. And the linoleic acid tallow enriched group were more severe than the lard ethanol group, but both of those groups showed histologic changes of fatty liver necrosis, inflammation in the liver. And their conclusion is our results strongly support the hypothesis that linoleic acid is essential for development of alcoholic liver disease in our rat model. Isn't that interesting? And this is not to say that you can just go out and drink alcohol with tallow and not get liver injury, but isn't it interesting that the majority of people in our society are now eating diets that are very high in linoleic acid from vegetable oils that I've talked about before, corn, canola, soy, sunflower, safflower, etc., peanut, and eating animals like chickens and pork that are fed corn and soy, and their fatty tissue is enriched with linoleic acid. And a lot of people that sadly have alcohol addictions do develop alcoholic liver injury. But if they were drinking alcohol and had an ancestral diet that was exclusively or mostly based on saturated fat from red meat, like I talked about with Nina Teicholz, maybe they wouldn't even get that alcoholic liver injury. What an interesting idea. It could protect people, but really the takeaway here is linoleic acid is probably not a good thing to get in excess. And I will talk about later on in this video what I think excess linoleic acid is, but there are many models that suggest excess linoleic acid is a very bad thing. Now, next paper, super interesting. dietary linoleic acid elevates endogenous 2-AG and anandamide and induces obesity. These are actually endocannabinoids. So these are molecules that are similar to cannabidiol, cannabidivarin, other cannabinoids that people might be exposed to from CBD, which CBD is cannabidiol, or marijuana. And we know that these endocannabinoids these are cannabinoids that are made in humans that look like those exogenous cannabinoids like CBD or cannabidivarin. And these have an appetite stimulating effect in humans. This is done in rats as well, but you can see here the dietary linoleic acid elevates endogenous 2-AG and anandamide and induces obesity in this mouse model. So basically what they're saying here is mice were fed diets containing 1% of energy from linoleic acid, 8% of energy from linoleic acid, and 8% of energy from linoleic acid plus 1% from EPA, which is an omega-3 fatty acid with some docosahexaenoic acid that I talked. Those were shown on the first slide. Those are omega-3 fatty acids. So the conclusions were that increasing linoleic acid from 1%, which is probably, at least in humans, again, this is a mouse model, but at least in humans, 1% linoleic acid is probably more ancestrally consistent to an 8% level elevated these arachidonic enriched phospholipids. They're saying that when you give these mice linoleic acid, it's a precursor for arachidonic acid, and that could induce endocannabinoid hyperactivity and promote obesity. So that's what it did when you jump from 1 to 8. This is akin to what's happening in the standard American diet. And it did that, arachidonic enriched phospholipids in liver and erythrocytes. It tripled 2-AG, which is one of these endogenous cannabinoids, and AEA and the anandamide with increased food intake, feed efficiency, and adiposity in mice. That's pretty wild. So giving these mice more linoleic acid was a problem. Now, interestingly, they say that the adipogenic effect of LA can be prevented by consuming sufficient EPA and DHA to reduce arachidonic enriched phospholipid pool and normalize endocannabinoid tone. This probably goes back to something I was talking about with Ivor Cummins, that if you are eating a ton of linoleic acid, that's not a good thing, but that may require more omega-3s. Now, the omega-3 to omega-6 balance seems to be important in humans, and this is probably one of the reasons that it is, because if the linoleic acid gets much higher than the omega-3s, the EPAs, and the DHAs, you're going to get these arachidonic acid-enriched phospholipids. You're going to turn on this endocannabinoid system. You're going to get obesity. And it's pretty interesting, as I'll show you in this article, they actually showed visceral fat increase in, actually in another article, I'll show you visceral fat decreasing in mice that were fed stearic acid, which is a saturated fat. So that's coming in a moment. But if you're eating lots of linoleic acid, you may need more omega-3 to balance that, but that's going to increase your overall polyunsaturated fatty acid pool, which could be problematic in general. The takeaway here is linoleic acid in excess in mouse studies, bad across the board, not a good thing. And it's been shown in human studies too, but I think these are interesting models. Let's talk about a few with stearic acid, which is sort of a complementary saturated fat. Now, as you all know, I did a podcast last week, Nina Teicholz, kind of exonerating, defending saturated fats, defending the saturated fats that occur in animal foods, specifically the long chain saturated fats like stearic acid. I will show you guys a profile here. If you can actually look at the profile of fatty acids in grain versus grass-fed cattle across the board. C18 is stearic acid. You can see that there's 13, 14, 15% across these different types of beef of stearic acid, suggesting that stearic acid is pretty well represented in grass-fed and grain-fed meat. I've talked about that before. Ruminants are unique in that they don't really get fat that is enriched with polyunsaturated fatty acids in the same degree as monogastric animals like chicken and pork. But this is interesting. What you will notice also in this paper looking at the fatty acid profile of ruminants is that the other one that we're looking at that's problematic is C18, 2, and 6. So this is linoleic acid, and we can see here pretty small amounts and smaller amounts in grass-fed meat. So I'll give a plug for grass-fed meat. You guys know I love this. More stearic acid and generally more stearic acid and less linoleic acid across the board in grass-fed meat. And you look at these percentages, linoleic acid, 1.93%, 1.69%. That's probably, in my opinion at this point, an ancestral level of linoleic acid in our diet. When we go much above that, we're going to cause metabolic problems, I believe. So let's look at some stearic acid stuff. So this is a saturated fat that most of the world would vilify. And you see a diet enriched in stearic acid protects against the progression of type 2 diabetes in leptin receptor deficient mice, DB mice. There are all these different mouse models that have to be sort of genetically modified to give them diabetes, but you can give these mice excess stearic acid, this is that C18 saturated fatty acid, and it protects against the development of diabetes in these. This is a dissertation from Valerie Reeves, super interesting stuff. And I'll show you one more study that shows pretty much the same thing, but the idea here is that this saturated fatty acid, this 18 carbon saturated fatty acid, is very important for signaling in humans. So I'm going to get to a human study as well, but dietary stearic acid leads to reduction of visceral adipose tissue in athymic nude mice. Athymic nude mice are the mouse model that is used for human breast cancer metastasis. And interestingly, as they show here, stearic acid is a long-chain dietary saturated fatty acid that's been shown to reduce metastatic tumor burden. And there's growing evidence that visceral fat is related to metastasis and decreased survival in breast cancer models. So what did they find here? They found that visceral fat in this mouse model of metastatic breast cancer was reduced 70% in the stearic acid group compared to other diets. In contrast, the total body fat only slightly reduced, but the lean body mass was increased in the stearic acid group fed compared to all other groups. Conclusion, unlike oleic acid, which is omega-9, this is basically what's in olive oil, and linoleic acid, which is, again, the 18, 2, and 6, this is the omega-6 that we're worried about. Stearic acid caused increased programmed cell death in pre-adipocytes. So it almost caused loss of visceral adipose tissue cells, which is probably what we want. And the thing I want to show you guys that's really cool here is they actually have pictures of the viscera of these mice. And you can see that this is visceral adipose tissue on a low-fat diet, high stearic acid. These mice had a six-pack. There's almost no visceral fat here. Corn oil, this is enriched in oleic acid, and safflower oil enriched in linoleic acid. Both of these mice had way more visceral adipose tissue than the mice fed an 18 carbon saturated fatty acid that is rich in ruminant animal fat. How cool is this? You guys may have heard me calling out vegans from time to time and saying, I would love to debate you and show me what your visceral adipose tissue is, because though I will get into the weeds and debate vegans and we can do all these studies back and forth and talk about this stuff, I also want to see what the visceral adipose tissue of someone is, because that is an indicator of their metabolic health. It's really undeniable. I can say, show me your six-pack. It would essentially show me the same thing, but getting an MRI or a DEXA to show visceral fat will really level the playing field. And I think what we'll see, as I will show you in this next study, is that those who are not eating ruminant meat and fat, stearic acid, are going to have more visceral fat, and that vegans are going to be metabolically unhealthy. How can I possibly make that claim? Based on the mouse studies showing that when the mouse are fed safflower oil, which is linoleic acid enriched, or corn oil, which is oleic acid enriched, their visceral adipose tissue got bigger compared to stearic acid, which is in that saturated animal fat from red meat, right? So does it happen in humans? I think it does. Check this study out, you guys. This is amazing. So this is actually done in humans. Dietary stearic acid regulates mitochondria in vivo in the body in humans. A couple people were saying, based on the stuff that I was talking about last week, how can he say this happens in isolated mitochondria? This is in a human body in an actual intervention. What the researchers did in this study was they put people on a low-fat vegan diet for 2 days. They saw levels of stearic acid decrease, and with it, they saw a decrease in mitochondrial activation, which they can assay by looking at mitochondrial fusion. When mitochondria are fused, they are working, they are doing beta oxidation, they are burning fat, and that is when mitochondria are turned on. What they found was that when they deprived people of stearic acid by 2 days of a low-fat vegan diet, the mitochondria fragmented and they became less likely to burn fat. And then they gave them stearic acid as 26 grams of stearate in a shake. And then they gave them stearic acid by itself, and they saw the exact same thing with very high numbers. The study was small, I think it was 10 people, but they reached clear statistical significance with a very small sample size. You could see that when you gave these people who've been on 2 days of a low-fat vegan diet stearic acid back, their mitochondria turned right on, and C18 increased fatty acid beta oxidation in vivo. They say, we show here, C18 ingestion rapidly and robustly causes mitochondrial fusion in people with 3 hours of ingestion. It causes a drop in long-chain acylcarnitines, that's carnitine, that's moving into the mitochondria. That is an indicator of beta oxidation, that the mitochondria are burning fat when you give them C18 in these people who had been on a vegan diet. Super interesting. They did this from neutrophils, and this is in vivo. So this is a cool diagram. You can see fragmented, intermediate, fused. This red stain right here is the actual stain of the mitochondria. They are merged after they have gotten the stearic acid. And if you read this paper, it's really, I think, worth reading the whole thing. They will say, as expected, the level of C18 in serum triglycerides dropped significantly after 2 days of a low-fat vegan diet. And the ingestion of pure C18, not in the banana milkshake, they did 2 experiments, induced mitochondrial fusion in vivo. I'll talk about palmitic acid more, but they show here that this is very specific with stearic acid, not induced by C16, which is palmitic acid, C18-1, which is monounsaturated, that is oleic acid, or C20. There's a dedicated pathway that senses stearic acid. Really, really interesting. And again, you can read here, it reduces acylcarnitine, which means it turns on beta oxidation. The mitochondria started burning fat, and it increased significantly after a 2-day low-fat vegan diet. Particularly striking, C18 leads to ingestion. Ingestion leads to an increase circulating C18 triglycerides, but causes a decrease in circulating C18 acylcarnitine, indicating that the acylcarnitine drop is not due to reduced substrate availability, but rather due to increased acylcarnitine usage. This is the movement of fatty acids into the mitochondria, burning the fat. This is turning on the engines. Interestingly, mitochondria were consistently more fragmented after 2 days of a low C18 diet in all subject groups: healthy volunteers, type 2 diabetics, vegetarians, and omnivores. This means that stearic acid, this C18 fat, is a signal that our body uses to burn fat. And I think this is super fascinating stuff, you guys, that this fat is so powerful, and yet these saturated fats are considered all the same, and we're not even, and this fat that is enriched in animal foods and is not found in plant foods is vilified. And yet, when vegans don't have it, their acylcarnitines are not being used, they're not doing fatty acid oxidation, their mitochondria are fragmented, they're not fused, they're basically not fat burning, their mitochondria are not working as well. I think the very plausible hypothesis here, based on that study and the rat or the mouse study, is that people who are not eating animal foods are going to have more visceral fat and are going to be more insulin resistant. So stearic acid, C18, good for decreasing visceral adiposity, which we know is when you have more visceral adipose tissue, you are less insulin sensitive, right? It's a bad thing. Linoleic acid and oleic acid, which is olive oil, remember that, increased visceral adipose tissue. So I've gotten a lot of questions. What about fish oil? What about olive oil? Well, at least in the mouse study, giving those mice oleic acid in corn oil increased adipose tissue in the viscera. So the jury is still out on monounsaturated fatty acids. I don't think they're horrible, but I would not make them the full part of your diet. What we are learning here is that lipokines, meaning that fats can act as signals in the human body. I'm going to talk about this paper more in the future. This is actually super interesting, the fact that our visceral fat can release a lipokine, meaning a fatty acid molecule that acts as a hormone to the rest of the body. This is a molecule that I haven't talked about in this little mini podcast, but I have talked about on previous podcasts with Cyrus and Robbie when I was debating them. They're fruitarians, but this is actually showing the same thing. And you could probably show the same thing with stearic acid, that these fats have hormonal roles in the human body. This one is specifically saying that lipid profiling led to an identification of C16-1N7, which is called palmitoleic acid, which is a different one that I've talked about, as an adipose tissue-derived lipid hormone that strongly stimulates insulin action and suppresses hepatosteatosis, meaning fatty liver. This is probably the fatty acid that our lipid-containing cells in our adipose tissue in our bellies are releasing to signal to the rest of the body to remain insulin sensitive. I'll tie this all together in the future, you guys. As I talked about in the first video on PUFAs, which was really, people were interested in this, I think that what's happening is that it all starts in the visceral adipose tissue. And you want to shrink that visceral adipose tissue. You don't want that to grow. And that shrinks when the visceral adipose tissue is insulin resistant. How do tissues grow? It's when they're insulin sensitive. So stearic acid might actually be making your visceral adipose tissue insulin resistant. It shrinks like we saw in the mice, and then it sends out a signal to the rest of the body with that palmitoleic molecule, palmitoleic acid, and that tells the rest of the body to be insulin sensitive. That is a lipokine. It's super complicated stuff, but it's really interesting. So what did we talk about? We talked about linoleic acid making rats have fatty liver and alcoholic liver damage. We talked about linoleic acid making rats fat, potentially making humans fat. We talked about stearic acid making humans, or at least mice, skinny and decreasing visceral adipose tissue. We talked about corn oil and safflower oil making rats fat and giving them more visceral adipose tissue. And we talked about how depriving humans of stearic acid, those 2-day low-fat vegan diet, led to fragmentation of the mitochondria. Connecting the dots, I have a real suspicion that if we took my visceral adipose tissue and compared it to a vegan's, they would have way more than me. If you guys have seen my abs, I clearly have abs. I don't have really any visceral adipose tissue, but show me the visceral adipose tissue of a vegan doctor, dollars to donuts. It is way higher than mine because they're neglecting eating important molecules that are healthy and found in red meat. Don't fear the saturated fat in red meat. Remember that grass-fed meat has more stearic acid, less linoleic acid, and don't get excess amounts of linoleic acid in your diet. All right, super interesting stuff. I want to close by talking about a few more things that are sort of related to this and sort of not. We will transition. Like I said, I'm doing a podcast tomorrow. Rob Wolf, Diana Rogers, we talked about cow farts and cow burps and how we've all been so misled. I got sent this article by someone and they asked me to please debunk the Guardian article recently, which says there are 18 reasons why you should not eat meat. This is Damian Carrington. I respect you, Damian, but you're way off base here. We went through many of these in the podcast. If you want to hear the response to this Guardian article, listen to the podcast with Rob Wolf and Diana Rogers. Diana was kind enough to share this little tidbit of information with me. Oh, by the way, the Guardian last year inked an $886,000 deal with OPP. This is not the regular OPP that you're thinking of. You guys are thinking of the 80s hip hop song. This is the Open Philanthropy Project, not the other OPP, which is a plant-based interest group. So isn't it interesting that the Guardian inked a $900,000 deal with a plant-based interest group to write articles that paint animal agriculture as inhumane, unhealthy, and dangerous to the environment? What the heck? How can we trust the Guardian? Geez. And a lot of the points that are made in that Guardian article are untrue, and you should listen to the podcast, which will be out tomorrow, and I will tell you all about why that is the case. So, last part, coronavirus. The news media is freaking out right now and saying cases are rising, but is it really true? It's not. So this is a really interesting little article that was sent to me by my buddy Anthony Gustin. This is June 29th, tracking COVID cases, hospitalizations, and fatalities. I believe this is in Florida. You can see that there's this, oh, look, all these new coronavirus cases. But the correlation, the connection with hospitalizations completely has broken down. And with deaths, completely broken down. In fact, deaths are going down. And if you look at the numbers across the United States, deaths continue to decline. So this correlation, this connection between new cases and hospitalizations or deaths has completely been fallen apart. And yet the media will not stop talking about this because they have nothing else to talk about. They have nothing else to talk about. So just crazy stuff, you guys. So that was super fun. You guys know how this is for me. I get a bunch of stuff in my brain. I have to talk about it. I have to share it with you guys. Hopefully you found that valuable with regard to the PUFAs and the stearic acid. Do not fear these things. I'm going to do a bunch of stearic acid experiments myself. My body comp is pretty good right now. If you see my Instagram, I've got a pretty good six-pack rolling in Texas right now, but I'm going to try and decrease linoleic acid a little more, play around with it. I'm going to have Brad Marshall from the Croissant Diet on my podcast later. Thanks to him for all of his sort of talk. We've had a lot of talks. We're still talking about this stuff, sharing ideas. Thanks to Peter at Hyperlipid, who I'm getting a lot of information from as well and trying to connect the dots in a unique way for you guys. So hopefully you guys like this. Check out my book, thecarnivorecodebook.com. Check out tomorrow's podcast with Diana Rogers and Rob Wolf. On the Fundamental Health Podcast, you can go to sacredcowinfo.com to pre-order their book. Stay radical, you guys. It's my birthday. I'm going to go celebrate. I'm going to go jump in a cold plunge and a sauna. See you guys.